I want to thank everybody for coming back. Our webinars, we took a little hiatus in July for vacation, and now we're back into the thick of things. Our talk today is with Noah and Malia from the Danforth Center, and they'll be talking about interactions between phenomics and database communities. But before that, I do have some announcements. Our RCN from the National Science Foundation has been officially funded. We're really super excited about this. The steering committee is starting to get things rolling again. You're going to see a series of emails, and there's just a lot in the background going on right now, but we just got the official notification that we can share with everybody that just yesterday. So I'm happy to tell everybody we are funded, and now things really get moving with agricultural data. I would like to introduce our speakers. First, Malia. She had her PhD in plant biology at Michigan State University Plant Research Labs. She focused on freezing tolerance, actually something I also did during my master's, which I started diving down into her PhD papers, and then worked as a postdoctoral fellow at Danforth Center, working on developing tools for high-throughput phenotyping, and is now actually an assistant member and a principal investigator at Danforth Center, continuing her work on high-throughput phenotyping, focusing on improving resistance to temperature stress in plants. Our other co-speaker is Noah. He is the director of the data science facility at Danforth Center. He has developed a number of tools, including one he'll discuss today called the Plant Computer Vision, and has a lot of information and a lot of useful user guides and help desks and GitHubs available for anybody who's really interested in learning more about the data centers, data science facilities work and future collaboration. And with that, I'll give it back to Malia.
Thanks, Jacqueline. Malia and I are going to introduce the Plant CV project, and we're going to kind of quickly go through some of the stuff it can do, which isn't completely always relevant to I think the interaction with the database community piece, but at the end we have some maybe discussion points we can bring up, and we won't take up the whole time with a talk, so there should be plenty of opportunity to discuss things, including things that Malia and I haven't thought about that you all have. So
yeah, we're really excited to interact with this group. So the title of the talk that we have right now is What Phenotypes Matter, sort of open challenges in plant phenomics with Plant CV, which is the software project that our groups develop. So the reason why I personally am interested in plant phenotyping is that I have a research interest in the relationship between stress and time, like Jacqueline mentioned. So if you give a plant stress treatment in the morning, you can get a very different physiological response than if you give a plant a stress treatment in the evening. If you give a plant a stress treatment when a plant is a juvenile, you can get a very different response in a plant later in development. If you give a plant a long progressive stress treatment, that can be very different, have a very different phenotypic response than a short acute stress treatment. And then the time after you phenotype a plant matters. So if you give a plant a stress treatment and then immediately phenotype it minutes or hours later, that can have a different phenotype than if you phenotype that plant hours or weeks or months later. So because of that very, very interconnected relationship, my lab and many other labs, I'm sure many on this call, are interested in non-destructive methods to assess plant stress and plant phenotypes. So at the Danforth Center, we were really lucky that they invested in technologies that allow for high throughput phenotyping. So in particular, this system on the left side here is a Lemnitech Scanalyzer 3D that we call the Bellwether Phenotyping Facility. So this is a conviron growth house that can house 1,140 plants, and they are on this conveyor system, and they can send those plants for daily imaging through RGB and near-infrared imaging stations. There's also 5 watering and weighing stations so that we can do very controlled nutrient application for, say, nutrient stress experiments, or drought or controlled watering experiments. Also in my group, we're interested in sort of new or different imaging modalities. So my lab has a hyperspectral phenotyping system. This is, it's hard to tell the scale in this image, but that is a 7-foot arm with a sort of 7-foot reach. So an industrial scale KUKA robotic arm. It's the kind of arm they use in the automotive industry to build cars. And attached to that arm is a very expensive hyperspectral camera. So we can do things like sort of spectral imaging. And the reason why we wanted to attach that very expensive camera to an arm like this is because the camera is actually more expensive than the arm itself. So we can actually image plants of different architecture types. So either side view imaging or top-down imaging, the same camera system. In addition, sort of these expensive commercial platforms, Noah and I sort of, when we first started our phenotyping careers, I guess, became interested in democratizing or lowering the cost of phenotyping. And so our labs are really interested in phenotyping with Raspberry Pi cameras. So these $35 computers that you can get $25 camera attachments to. And with these, we can build or outfit growth chambers with lots and lots of cameras so we can collect image data at very high temporal resolution. So with all these different phenotyping platforms, and these are only a few that we kind of have at the Danforth Center, our problem quickly switched from data collection to data analysis. So we needed to be able to extract numerical trait information from all of the different imaging types that images and imaging types that we were collecting. And I should mention that the Danforth Center got their phenotyping system back in 2013. And so at the time, there were sort of fewer options in terms of open source projects to analyze image data. So what we had commercially available to us with the commercial platform was software that was slow because we had to pay to put for sort of every CPU we wanted to put that software on or use. And it was sort of a black box. So we had no control over how traits were defined. So just as an example of this, the software developed by software engineers, and we love software developers and software engineers, but they don't necessarily have domain knowledge in plant science to be able to define plant traits in a way that plant scientists might. So the software engineers defined plant height as this orange line, and that's because there's plant material sort of at the top of the line and as well as the bottom of the line. But that's not how a plant scientist might define the height of a plant. And there was no way for us to sort of modify or have input into this software until we quickly realized that we needed different software tools that were flexible and that could be clearly defined for research as well as to ease publication. And so that's when we really decided to develop the project that we call Plant CV, which stands for Plant Computer Vision. So the vision of Plant CV is to build phenotyping tools that'll be modular and reusable so that they can be combined and recombined with ease, so that we could build flexible workflows to quickly and efficiently extract biologically relevant data from both images and sensors. These tools needed to be documented well enough that they could be usable by both bioinformaticians, data scientists, and biologists. And it was important to us because as biologists, we wanted these tools to be accessible to labs that don't necessarily have access to computer scientists or active collaborations with computer scientists. So at its heart, Plant CV is a library of modular functions that you build into workflows. And these workflows can be simple. So if you have a very simple image, say a plant on a perfectly white background with no shadowing, then that workflow can be very, very simple to extract the plant material from the background or segment the plant material from the background. Or it can be more complicated if your image data is more complicated. Once you have a workflow set, that workflow can then be parallelized over a set of similarly captured images. So then you can extract and result the numerical data can be extracted and result in a database of measurements associated with metadata. We never wanted to develop PlantCV in a vacuum, so wherever possible, we try to be interoperable with other open source image analysis packages like scikit-image, OpenCV, SciPy, just as some examples. We do not want to reinvent the wheel wherever possible. If you're interested in a lot of the details concerning PlantCV after our talk, then this is a link to our website that also links out to our GitHub. And if you're interested in sort of version 1 and version 2, those are 2 publications that are both listed below. So one issue or problem that we saw back when we first started developing PlantCV is that there was a lot of people in the phenomics space that were dedicating a lot of personnel time and resources to developing really innovative algorithms, important algorithms, and also software. And they were doing that in the research space with graduate students or postdocs who eventually move on, graduate to their next steps. And so a lot of that, those tools and resources sort of died along with that loss of maintenance. And so a lot of what we're hoping to do with PlantCV is to build infrastructure that allows PlantCV to be sustainable over time. And so a lot of that time and effort that the core PlantCV development team dedicates, we dedicate our resources towards is building infrastructure. And so, like I said, I think previously, PlantCV is available through GitHub. We allow contributions from outside contributors, and we are able to do that because of the infrastructure that's sort of built in. So we're compatible with projects like CodeCov, which allow for code testing. What that means is that if I build a function that breaks something that Noah's previously contributed to, I can't push that function into PlantCV without those merge conflicts being resolved. We spent a lot of time, well, Noah in particular and Haley have spent a lot of time making sure that we have nice installation available through PyPy, Docker, and Condaforge, and sort of monthly or continuous or monthly releases, so that there's open development of PlantCV. Our documentation is currently available through Read the Doc. So every function in PlantCV has documentation, and all of the functions also have interactive documentation. What that means is that you can test to see whether or not PlantCV will work for your particular image data before you go through the effort of actually installing it. This is an incomplete list of the features that are available in PlantCV, but just some notes. We can currently process RGB grayscale, near infrared, PS2 or fluorescence imaging, thermal image data, as well as hyperspectral data. Within the package are sort of all of the classical computer vision methods that you would need to segment an image. That we have graph-based segmentation methods, which Noah will talk a little bit or highlight in a bit. There's a number of new data visualization methods. There's machine learning methods, which we'll highlight as well. And then there's a number of different measurements that can extract through PlantCV. We're really proud of the PlantCV community that we've built over the past several years. So like I think I said, we got the Bellwether phenotyping system in 2013. We started developing at the fall of 2013. We started developing PlantCV in 2014, and the official release was in 2015. So we average about 30,000 downloads per year via PyPy. We have 30 contributors to PlantCV, and what's really nice about that are many of these are community contributors, so not people who are directly involved or directly paid off of sort of PlantCV grants, associated grants. We've had over 4,000 commits to the Plant CV project. And there's been 5 papers on the direct development of Plant CV, as well as 31 publications from people who have used Plant CV in their research. So these are people who are not necessarily from the Danforth Center, but are using Plant CV as a tool. And that's stats that we're sort of really proud of. 11 of those papers have actually been published as preprints or full publications in this past year. And so you can kind of tell by that the number that we're kind of growing in the number of people who are sort of using Plant CV. We find that Plant CV is a really useful tool for education and outreach. And that's because, especially for people learning or beginning bioinformatics or data science. And that's because traditionally we were using genomics as a way of teaching bioinformatics, learning to analyze RNA-seq data or assemble genomes. But what's difficult with that is that you need to have it's not as visual, and you need to have some sort of base knowledge about genomics in general. We find that it's a lot easier to explain to younger researchers, especially people in the K through 12 space. Our goal is to segment the plant from the background of an image. And so we often use Plant CV as a tool, again, when we're using in these education and outreach projects. It also helps that we have Plant CV integrated with projects like Jupyter Notebooks, so that, again, people can get visual feedback on all of the steps in Python that they're kind of going through. So next, Noah is going to highlight some of the newer features that have been added to Plant CV in the past year and a half or so. And then we'll move on to sort of some discussion points that have been put together.
Yeah, and I can kind of go quickly through this because a lot of the specific details don't really matter, but just to highlight a few things that might be generally interesting. One of the major things we worked on over the past year or so, Haley Scholl, who's a developer in the group, did most of this, was developing an entire submodule for this graph-based segmentation, particularly for grass plants like you're seeing here, where you can we can treat the stem and the leaves as primary and secondary segments in a graph structure, and then segment them into leaves and stems based on whether they are primary or secondary branches. So if you go to the next slide, then given that we can segment those paths, we can then measure some interesting plant phenotypes from that. So things like the length of each leaf, the insertion angle with the stem, the leaf curvature, and a bunch of other properties that are interesting to be able to do in high throughput. So next slide. Right, so some of the machine learning stuff that we've worked on is for either classification of plant pixels or segmentation of images into multiple sets, multiple classes, as we would call them. And so in the simple case, in this case, we're just trying to separate the plant from the background. And so we can do this by color, by building a statistical model of what plant colors look like versus the background colors in this image. And then this model, once it's trained with actually not very much data really, can be used on additional images. And so for a more relevant example of how this has been used on the next slide, Malia was working with some folks at University of Minnesota. Is that right, Malia? Yeah, thanks. So they had a bunch of scans of wheat leaves with rust infection, and they wanted to identify the orange pustules and the chlorosis on the leaves, as well as the leaf area, so they could estimate how susceptible the plants were, and they were doing this by like a qualitative scoring method before. So we used this naive Bayes classification system where you can select like color values from different regions of the image and bin those into classes. So on the next slide here, we build a table of color values for each of the classes you want to define, and then that ends up being the training data for the model. And on the next slide, you'll see can be used to classify at the pixel level which group the pixel belongs to or is most likely to belong to. And so then you can use that model to make quantitative predictions for the whole data set, which has worked really nicely. And the good thing is it correlates well with the qualitative measurements that were being done before. And then we've used a good thing for us is we've been able to use the same tool for different applications, which is nice. I mean, it kind of gives us some validation that we didn't just make a tool for one-off use case, but it's been useful for other projects. And this example is from the Springer lab, also at University of Minnesota, where they were using this tool to identify parts of the plant that I forget the terminology. Did they call them necrotic? Yes, necrotic pixels, because these plants are being exposed to cold stress, and so they're kind of trying to measure the effects of cold stress based on unhealthy parts of the plant that they're sort of classifying with this system. So they did this for a whole bunch of maize inbred lines, with cold and not cold treatments. And then one more example of this, we worked with the Carrington lab where they were infecting Arabidopsis plants with turnip crinkle virus. And so we used the naive Bayes classifier to do something similar where we can like separate the plant into both like predicted uninfected parts and infected parts based on visual assessment of whether it looks green or not green. And then we're also showing here on the graphs on the right that in addition to classifying them as either healthy or unhealthy, you could instead also just use a quantitative measure of the color of pixels to assess disease severity. So the plants in the mock column, you can see they always stay green, as healthy plants do. But in the middle column, for instance, you can see a shift for most of the plant pixels from green to like yellow orange when they become severely infected by the wild type virus. Cool. Next slide. So Malia mentioned that in addition, to the sort of the big commercial phenotyping systems, we use a lot of Raspberry Pi cameras to do affordable phenotyping. Malia's group operates, I think, 200 or 300 Raspberry Pi cameras in total across 5 or so growth chambers, I think. So it ends up being quite the infrastructure, but she can image thousands and thousands of plants simultaneously across those facilities. They have some downsides, though, of course, to the commercial systems. So one of them, the biggest one, probably being that we are not imaging individual plants, in a perfect, in perfect isolation like we are in the commercial system on the conveyor belt. So one of the challenges is, well, how do we separate the individual plants and measure them individually? So Jorge joined our group just in the middle of the pandemic, so I think he's never even worked at the Danforth Center on site yet. But he has a computer science background, and so he's brought in a lot of interesting ideas like to that team, and he tackled this problem of how do we segment individual plants from these more complex scenes. And so in the picture on the left, when the plants are young, that's pretty easy because they are separate from each other. And so we can segment them individually, and we can treat what we would call connected components as one plant. And then in the next picture, once they start touching each other, those connected components now span multiple plants, so we can't tell them apart. But his idea was to use the initial individually segmented plants as markers, because we know that they're separate, and then stack the images as a time series, and rather than treating them as individual pictures, and use a watershed algorithm in 3D where the third dimension is time to propagate the labels over the time axis. And so it's actually pretty successful at identifying the boundaries of individual plants, even when they're touching or overlapping. And so, that allows us to extend the period of time where we can measure individual plant phenotypes in these more affordable systems. And then Hudan-Yun Sheng in Malia's lab has also joined the group in the last year and a half, and has also made a ton of contributions to the project. And so one of the big things that she tackled was this project with deep learning. And I won't go into the details, but we're using a tool called MaskRCNN, which was trained on synthetic Arabidopsis pictures, and those citations are in the bottom right, to do leaf segmentation. So the synthetic training data works really well at training the model, even though it's never seen our data at all. And so in the middle video, the strobing you can see is the labels being applied to the individual leaves at each time point. And so you can see MaskRCNN just arbitrarily assigns an identifier to each leaf. So we know where the individual leaves are, but we don't know from one time point to the next which leaf is which. And so what Hudan-Yun worked on is a time series linking function that uses an optimal assignment algorithm to look at the overlap between leaves between time points and make an assignment of which leaves are likely the same leaf between time points. And then by doing that over the whole time series, we can apply a uniform leaf identifier to each leaf, which is why you're seeing the colors remain consistent over time. And then because we can do that, we can measure the phenotypes of individual leaves in this case over time, and measure things like leaf initiation rate, leaf size and shape, color even, and all sorts of things. Like these kind of time series based approaches like open up a whole nother sort of avenue for phenotyping that we didn't have before. Cool. You go to the next. Back to you, Malia.
So one thing that we wanted to highlight just at the end was this project we had in quinoa. And that's because when we were first getting started and sort of talking to a lot of people in the phenomic space, the consensus was, or the idea was let's just collect all the numbers and all the phenotypes and extract all the information. But sometimes you need to have some idea about what phenotypes matter before you go into a project. And so we kind of wanted to highlight some of the thought that goes into narrowing in on what phenotypes matter and how to sort of figure that out. So we have, in my group, this project on heat stress in quinoa. So we had 4 treatment groups: a treatment control temperature treatment group, a treatment where we specifically only heated the roots, a treatment where we heated both the roots and the shoots, and a treatment where we heated the shoots only. And we were interested in this differential heat stress treatment because quinoa is known to have a very ideal sort of nutritional profile in its grain. And so we were interested, but high stress tolerance. And so we were interested to see if there were differences in these different heat treatment groups. So when we applied heat treatment, we started at a thesis. The heat treatment lasted for 11 days, and then we returned the plants to controlled treatment temperatures. We didn't know what to expect in terms of yield after these heat treatments. So in quinoa, we see that anything with a heated shoot treatment, so both the heated roots and shoots and the heated shoots alone, had a significant hit in yield in comparison to both the controlled treatment and then the heated root treatment. This is not necessarily what we knew would happen or expected. So if you specifically heat roots in wheat, you can get actually a very opposite phenotype. So we were interested in knowing what contributes to this yield loss in this heated shoot treatment. So I'm condensing the story a lot, but one of the things we did was collect RNA-seq data. And we saw that anything that had a significant, in the treatments that had a significant yield reduction, there were about 400 commonly differentially regulated genes. Of those 400 genes, there were 10 transcription factors, and pretty much all of those transcription factors had something to do with flowering. And so because of that, because of this particular phenotype, we decided to look at flowering in quinoa. So one interesting thing to point out for one of those genes is that for Arabidopsis, when you have a downregulation of ARF2, which is one of these differentially regulated genes, you see a yield hit because there's a change in flowering opening and closing. And so because of that particular phenotype, we started to look at flower opening and closing in quinoa. And what we saw was that with heat stress with the heat stress treatments that had a significant yield reduction, there was also a change in the length of time that they had open flowers during the day. So our hypothesis is that there's a reduction in yield because there's less time that those flowers are open, and there's less chance for those flowers to be openly pollinated. So ideally, we would take this phenotype and phenotype it across a larger population, say an accession population or a recombinant inbred line population. But this particular phenotype is really difficult to assess manually. So you could manually count flowers. That's how we initially assess flower opening and closing. But we're actually interested in using different imaging modalities to see if we can estimate this phenotype more easily. So when flowers are closed in quinoa, the anthers sort of bunch together, and when the flowers are open, they spread apart like this. And luckily, or hopefully, we think luckily, the anthers of the plants can easily be spectrally separated from the rest of that panicle material. And so we think we can maybe separate or distinguish, estimate open flowers versus closed flowers based on the size of that estimated anther size. So just as a summary of this portion before we kind of go into the discussion section, that is the website for PlantCV. We always encourage suggestions and contributions at any scale to the project. I don't think we mentioned this, but for PlantCV version 2, a lot of the authors on that publication were sort of anonymous or people we didn't know initially in terms of, and they were contributors to our sort of GitHub, through GitHub. So PlantCV is interested in sort of tools that lower the cost barrier for high-throughput phenotyping. And we're working on tools for 3D watershed segmentation, as well as time series linking and new hyperspectral tools and thermal analysis tools to hopefully extract more information from different imaging modalities. So some of the open challenges we have in the lab are problems that are specific to our labs are how we quantify changes in flower opening. And we're always interested in sort of these open challenges that labs have. So if your lab in particular has a challenging image analysis problem or trait that you're trying to extract, those are things that we're interested in collaborating on. So before we get to the discussion section, we wanted to thank sort of the core developers of PlantCV, Haley, Jorge, and Danny in particular, as well as many people that work on PlantCV in our groups. And particularly wanted to point out our USDA FACT funding that directly funds the development of an expansion of PlantCV. Okay, I'm going to hand it over to Noah again for the next slides.
Cool.
I guess
so. These are some things that are not solved problems for us that I think would directly relate to like interactions between like a tool like this and the database community. They may not be the only problems, or they may not be what you view as important, which is fine. And anyone who has ideas about, other things we can talk about, that's great for sure. But I'll just, as I'm going through this, if you have questions or want to like talk about something else, just stop us and we can go on. So this is just a little bit of background. So we talked about like image analysis workflows, and I'm just pointing out like a workflow for us is a directed acyclic graph. And so you plug functions together. So you've got the outputs of one function or the inputs of the next, and so on. So you can build a workflow as complex as you need to work on the data you have. So this is an example of a kind of a reasonably complex workflow on the right here, where there's two input images. There's an image with a plant, and then an image of an empty pot. And so this is kind of using a background subtraction approach to remove some of the scene from the image with the plant, as an example. So you build a workflow, based on some sample data from your data set. And then the next goal, I think if you go to the next slide, right? So we want to run that workflow in parallel because image data sets tend to be fairly large. So you may not want to run a workflow on all of the data within a data set. Maybe you have different imaging modalities. So we need to be able to specify which images we want to analyze. We have to extract metadata from the data set, typically in image file names, but possibly from elsewhere. And then we want to run each image through the workflow, again, in parallel. So those are sort of like the overall goals of what we are trying to do, and this part works for us mostly. But if you go to the next slide, so currently how we get image metadata, and I think this is one of the two places where I think there's consequences between how we get data and then like how that data links to other data resources later. So in this example, I'm showing some boring text of a workflow configuration that one might set up for analyzing a data set. And so I've bolded a couple of the configuration options. So the first one is called file name metadata. And so we have the user define for us how metadata is encoded in their data set, in their image names, for example. So I think if you click next, I'm just showing some made up file names here, where the file name is composed of a few metadata terms. So the like in the first file name, let's say it's side view underscore RGB underscore a timestamp of when the image was taken. So we can define the structure of the metadata as using the terms camera, image type, and timestamp. And then I've shown a couple other examples here of like metadata filters a little bit down below, where we could filter the data on particular on particular attributes. So we only want to analyze the image type RGB. If we had other data, it would be excluded. And we only want to analyze the side view camera. So the file TV RGB would be ignored in this particular analysis. So if you click next, what we've done to, I guess, make this somewhat consistent. I mean, this is really not well worked out, but something I think like would be useful to think about for our community and how these things would interact with other communities is the metadata terms. So we have a controlled vocabulary for what terms can be encoded in the file names, how we extract the metadata. So these terms are defined in our workflow configuration, and I'm just showing two examples here of how we define them. So camera, has a label, which is a human readable name of what that term refers to. The data type, like programmatic data type, the value is stored in, so it's a string. And then the default value or the inherited value once it gets set. And versus a timestamp is a date time string and not just a regular string. Yeah, so anyway, like I think like that's one place where yeah, like having like regularized terminology in terms of like how we encode metadata would be important and not something we've worked super hard on at this point. And then I think on the next slide, I think this could be taken a bit further, and we don't do this. And I don't know if I know many of you would be, of course, aware of other kinds of controlled vocabularies, ontologies, and minimum information specifications. But one in the phenotyping community that's important is minimum information about a plant phenotyping experiment, or MIAPI. And they developed their specification to map to the investigation study assay, or ISATAB sort of structured data set kind of specification. And like right now, we are not really gathering experimental metadata information in plant CV other than what's encoded in file or accessory sort of files that come with an image data set. But I think it would be really interesting to actually have this information as part of like running plant CV, because I think we could get information about like more about like deeper information about what's actually being analyzed, right? So if it's a I don't know, we could have like terms say, or we could link, like, I guess, the experimental information back to information about like what the organism is, or what organs, or other plant structures are being analyzed. So I think that could link to plant ontology and things like that, which would be, I think, interesting to make the sort of output data sets richer.
So if I may suggest, no, I think you're raising a very good point over here. Is there are different methodologies and different standards available from different communities, and sometimes the data that the plant breeders would generate in a QTL experiment, for example, would be a timestamp and environmental conditions probably at the end of the project, whereas what Malia is developing right now, or collecting the data, is as a time series, for example. So even though your trait and phenotype is being averaged over a period of time, you have environmental conditions from every data point that you collected in that series. So how does that fit into the MIAPI workflow to address? Because it's not a one-time shot, it's a time series, and how you go around it, kind of things. So that's interesting. And
good point.
yeah, any suggestions on that side?
I think it can. I mean, I definitely think that is. I think time series are a bit of they're a problem because I think we do tend to like, yeah, this experimental description is sort of a one-time thing. I think to some extent they can deal with it by like the observational variables and the observations themselves. Well, I guess the observational variables define what kinds of observations you collect. So like what kind of traits you're measuring. And then the observations themselves, I think, I think this is where PlantCV is plugging in, is generating those observations. So even though it's generating multiple observations of the same plant over time, I think it can still fit within this framework.
Yeah, because what Malia showed it in, I think in the very first slide, 4 different conditions, either a growth stage or you tweak one parameter and the phenotype result is completely different. So that would be a beauty of that time series to know. At least we have environmental parameters and the growth parameters from every data time point in that series, and then somebody replicates but tweaks something else changed. So I think that would be the critical to see what are the results of it and what is the most optimal time point where that should be given or treatment should be given or something like that.
Oh yeah, yeah, definitely. Great.
And the second one I was thinking about is when you're collecting the images in the PlantCV, is there a mechanism where you basically ask the researcher to start as a set up the experiment and what kind of data are you collecting, what kind of plants, what kind of mutants and species and things like that, so that some of the standard data that you're looking at in MyAPI is actually filled in as part of that experimental setup, and then you obviously use 5 different methods, so those become part of the phenotyping protocol mechanism. It's a total workflow in that sense, so that's everything is standardized for them.
Oh yeah, I agree. I think if you don't standardize the process up front, then yeah, I think it's hard to go back and ask people to fill in the information because they're like, well, the experiment's done. Yeah, and I think for like our facilities, that's definitely doable. Actually, our phenotyping facility director basically does this, maybe not in this standardized way, but she does collect standardized information, about what kinds of plants are being loaded and what treatments they're getting and things like that. So I think be an opportunity to use this method as for at least starting in those kind of facilities, because you kind of have that control up front.
Yeah, absolutely. Thanks. And I love the ontologies integration in that CV about what kind of environmental parameters and traits you're measuring. So if you can bring in ontologies in it, that would be fantastic.
Great.
I'd love to work with you guys.
Awesome. Yeah.
Yeah.
Yeah. Well, and then on the next slide, I think it comes back because, oh, well, okay, not quite. Well, anyway, we do analysis in parallel. And in the last year, we've added an ability to do this in a distributed way. So we use Dask for this. So Dask can act as an abstraction layer between lots of different kinds of infrastructure. So you can distribute PlantCV workflows on any of these kind of clusters. So next slide. So the other part was about the inputs, and then this is about the outputs. And so we've done a little bit more work here on trying to make, I guess, PlantCV outputs more standardized and maybe more compatible with downstream use. And so we've used in that paper that was linked to before, that was, I think, kind of like one of the first MyAPI papers. One of the things they described was, how to define observation variables and then how to, how to sort of the components of defining observation variables. So we use this mostly the way they specified. So we kind of, the overall PlantCV output has an observation variables section, which defines the kind of variables that were collected in an analysis. So they don't all have to be collected every time. The user chooses which analyses to run. So I'm just showing two examples here where we have a metadata type variable like the plant barcode, and then a measurement type, which is the area in this case. And then for each, I call it entities, but entity basically is an image. We can collect two types of things. So one is metadata. So this is passed from the inputs that we talked about. And then the second is observations. So the things we measure. So we have it set up. We don't need to talk about it, but you can collect multiple sets of observations per image. So that's what this sample one refers to. But for a given observation, we use like the sort of structure proposed by the MAPI group. And so we have a variable. I think if you click one more time, Malia. Thanks. So we have a variable ID that's just a unique identifier that is used to index the observation. And then trait, which is the human readable like name of the trait. The method, which in our case is which PlantCV function generated the observation. The scale, which is the units that the observation's in. And then we did, well, then the value, of course. And then we added two additional components, one being data type, because programmatically, I think that was useful. And then label, which has to do more with something I won't talk about, but kind of an additional parameter that we have. But anyway, so we structure all the outputs like this. And then if you go to the next slide, Malia, in our documentation, we link out to a table where I've worked on sort of mapping these observation variables to some vocabulary stuff and to sort of organize like around like, you know, the methods being used and data types and whatnot. So like as just an example, the pixel area of the plant, or it could be converted to like a metric units if you calibrate the data, you know, could link to, in this case, a pedo term, or the median hue value of the color value of the plant, can map those to a method, which is calculating the median, and also the hue as the attribute. And so anyway, there's things like that I think we could do more work on to sort of make the measurements more meaningful and interoperable with you know, other systems that we would definitely love to work with people on, so that like these phenotyping tools can work better with other systems. So yeah, I don't know if that's, you know, there's probably, of course, many other like things to think about in terms of like linking the phenotyping and database communities. But those are some of the things that have been on our radar as things that we need to, I think we need to work on our end to help with those interoperable linkages.
So one question, this is Pankaj here, is I mean, obviously we have a lot of data archives for molecular data sets, but not necessarily for the phenotype data, that raw data that you or the community is collecting. Have you thought about something similar to create a national or an international resource, or you know, or we should collectively go and talk to the funding agencies to see what can be done so that we can archive all this data and pipeline some of the analysis and reanalysis tools too, because some of the versions of the software also changes, so the analysis may change a little bit as well from time to time. Because it's an interesting data because you get only one shot to collect such a comprehensive data on a population. You don't do those kinds of things again and again, and so there might be some hidden traits that may be revealed later on from maybe 2 years down the road, which were not analyzed beforehand. So how do we archive this thing?
Yeah, it's a really good point. I think in our community, we focus a lot on sharing the image data, or the lack of places. Well, it's not that there aren't places to share the data, but there's definitely no centralized repository for image data. So we do talk about that a lot, but I also think what you mentioned is interesting too, which is not just the image data, but the phenotypes themselves too, and in some sense they're easier to share because they're much smaller than the image data.
Yeah.
But they do. I mean, as you can see here, they're also more complex to some extent too, because you could theoretically have people define like area, a number of different ways, or height a number of different ways, or the units could be different. And so there would be a need to figure out a way to, make sure we're measuring something similar. That I could imagine being a challenge. But yeah, I think it's definitely a challenge. I mean, maybe it's easier to tackle within specific communities, like through the database communities where you have, Maze GDB or, other sort of more community focused groups where you can sort of define, at least for that community, like what the standards are.
Mm-hmm. Thanks.
All right.
I think we're at our 1 hour mark, but Lenore just asked an interesting question. I'll bring it up as our last question. With respect to the Rhabdopsis data, there is a Afro something project from Arthur Knock. Have you looked into that resource?
Arafino
There we
project.
go.
Yeah, Arthur Court. So I know that he was working on some of the sort of experimental metadata and integrating that. I don't know what your interactions with that project have been. That's just, I know, a small subset of the kind of experimental data that you're capturing too.
Yeah, I know of the project, but I haven't looked into it that much. But I do think, I like the idea of, I like the idea of like working with like a group like that, where there is like where there is a specific use case, because I think that's just easier to, to do something like tangible, for sure. I think I remember, are they the project that I'm remembering? Sorry, just going to there.
I think, he was sort of trying to, to serve that, a bit of that, function of a repository, but I don't know how much uptake that it has.
Mm-hmm.
But yeah, I agree with the whole sort of concept that we need to really be able to structure, phenotype data in a way that, enables a lot of cross-species comparison, and also, that we have a generalized repository of all of this. Yes.
But this is, like everything else, it's really nice to work with, because for most of its life, we can actually kind of treat it as a two-dimensional plant. And so in the Raspberry Pi systems, for example, it just works really well, and we can do a lot of cool stuff with it that are much harder to do with, bigger plants, for sure. And so I think as like you know, like the other things that make it a good model system, I think it also is a good model system for working on some of these problems, because of that ease of use, for sure.
I want to thank Noah and Malia again for leading a really interesting topic. I mean, this has been really interesting and just helpful for us as a community to kind of figure out like how could we help, or what kind of data is going to be thrown our way from researchers like Malia. So it's good to start the discussion, and maybe we can have even future topics, maybe in a different revenue, maybe the new AI project that Darwin is, we're just now learning about, So
Yeah, definitely. If anyone if you think about it for a while and have some thoughts, we'd definitely be interested in chatting more, for sure. And thanks for the invite. Yeah, and congrats on the RCM.
Yeah, thank you. All right, well, with that, we'll go ahead and close the meeting. Thank you everyone for attending.